Hello everyone, welcome to the channel Microbial World. Today we will be learning about the process of glycolysis. Glycolysis is also known as EMP pathway. It is named after three scientists, Gustav Emden, Otto Meyerhoff and J. Pernas, who gave the scheme of glycolysis. It is a pathway of glucose catabolism where glucose is broken down to produce energy. The process occurs in the cytoplasm of all living cells, aerobic as well as anaerobic. All of the glycolytic enzymes are also found in the cytosol. In the process of glycolysis, glucose is converted into pyruvate. The complete reaction of glycolysis that occurs in the cytoplasm is as follows. Glucose plus 2 NAD plus 2 ADP plus 2 inorganic phosphate gives 2 molecules of pyruvate plus 2 NADH plus 2 ATP plus 2 H2O plus 2 H+. Now let's look into the steps of glycolysis in detail. The first step of glycolysis is the conversion of glucose to glucose 6-phosphate. The enzyme that catalyzes the reaction is hexokinase. Here the glucose ring is phosphorylated. Phosphorylation is the process of adding a phosphate group to a molecule derived from ATP. Hence, in this step, one molecule of ATP is consumed and ATP is converted into ADP. Magnesium is also involved in this reaction. So, the glucose ring is phosphorylated to form glucose 6-phosphate. The second reaction of glycolysis is the rearrangement of glucose 6-phosphate into fructose 6-phosphate by the enzyme phosphoglucose isomerase. It is an isomerization reaction and it involves the rearrangement of carbon-oxygen bond to transform the 6-membered ring to a 5-membered ring. The enzyme phosphofructokinase with magnesium as a cofactor converts fructose 6-phosphate to fructose 1,6-bisphosphate. Similar to the reaction that occurs in the first step of glycolysis, here also one molecule of ATP is consumed and it provides a phosphate group that is added on to fructose 6-phosphate molecule to form fructose 1,6-bisphosphate. The enzyme aldolase catalyzes the cleavage of fructose 1,6-bisphosphate to form two 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So here the first phase of glycolysis is completed, and as we have seen, energy was utilized in the first phase of glycolysis. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are isomers of each other. So the enzyme triose phosphate isomerase rapidly interconverts the molecule. Glyceraldehyde 3-phosphate is the only molecule that will continue in the glycolytic pathway. So all of the dihydroxyacetone phosphate molecules that are produced is acted on by the enzyme triose phosphate isomerase, which reorganizes dihydroxyacetone phosphate into glyceraldehyde 3-phosphate so that it can continue in the glycolytic pathway. At this point of glycolysis, we have two 3-carbon molecules. In the next step, two main events are taking place. Glyceraldehyde 3-phosphate is oxidized by the coenzyme NAD, nicotinamide adenine dinucleotide, and the molecule is phosphorylated by the addition of free phosphate group. The enzyme that catalyzes this reaction is glyceraldehyde 3-phosphate dehydrogenase. The enzyme allows NAD molecule to pull a hydrogen off the glyceraldehyde 3-phosphate, converting NAD to NADH. The phosphate group then attacks glyceraldehyde 3-phosphate molecule and releases it from the enzyme to yield 1,3-bisphosphoglycerate, NADH and a hydrogen atom. Phosphoglycerate kinase transfers a phosphate group from 1,3-bisphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. As there were two 3-carbon products from phase 1 of glycolysis, we actually synthesize two molecules of ATP at this step. In the next step, the enzyme phosphoglycerate mutase relocates the phosphate group in 3-phosphoglycerate from 3rd carbon to 2nd carbon to form 2-phosphoglycerate. The enzyme enolase removes a molecule of water from 2-phosphoglycerate to form phosphoenol pyruvate. The enzyme pyruvate kinase transfers a phosphate group from phosphoenol pyruvate to ADP to form ATP and pyruvate. Again, since there are two molecules of phosphoenol pyruvate, here we actually generate two molecules of ATP. The second phase of glycolysis completes here. As we have seen, energy was released in the second phase of glycolysis. In the first phase of glycolysis, two molecules of ATP were utilized to convert glucose to glucose 6-phosphate and also to convert fructose 6-phosphate to fructose 1,6-bisphosphate. In the second phase of glycolysis, 4 ATP molecules were synthesized. During the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate and also in the conversion of phosphoenol pyruvate to pyruvate. So the net ATP produced is 2 molecules of ATP. Thank you for watching. I hope that the video was helpful. Do subscribe the channel for more informative videos.